I will get 10 hours of sleep at night and I'm still exhausted.
Does it affect your blood sugar control?
Not really. Because my current pump is really, really good about compensating for me not eating. I can eat one meal a day and then it'll be fine. Like my blood sugar doesn't go low during the day, even if I literally don't eat until 9 p.m. Which is really nice because sometimes I don't.
Do you wake up high? No.
I wake up right where I'm supposed to be. I go to bed high sometimes because I am really bad about eating during the day. I'll eat a late dinner and then I'll go to bed. So I tend to go to bed high, but I come down really quickly.
You don't take nighttime insulin?
I take because I don't take a long acting, I only take short acting. So I take insulin for my meal, but you still have a spike if you're eating a lot. Because just how my depression is and how my schedule is, I like had my endocrinology appointment and she's like, it looks like, she's like, are you not bolusing through your meals? It looks like you're not eating anything till like 9 p.m. And I said, if it doesn't look like I'm eating till 9 p.m., I'm not eating till 9 p.m. I don't know. I'm a college student. I work all day. I can't always eat breakfast and lunch.
Yeah.
And a lot of times if I'm like, I'll have like a no carb snack kind of a thing, about cheese or whatever.
Yeah.
But, yeah. Super fun, but yeah, I rarely have nighttime lows. I love my closed loop system because if I start to go low, it'll correct it before I even go low. So I can sleep for 16 hours straight and not have to worry about my blood sugar, which is a big relief. How hard is this quiz going to be?
Not hard. Ah, man. Get that to work.
There you go.
That's up as high as it'll go.
I can hear it. And Jenna should be able to do what we're close enough.
Man, that hum. My computer was turned down too low. Okay. Ooh, that buzz. Yikes. Ooh. All right, so that should work. Okay, Dane, come on, buddy. Hey, Sarah. Stay warm.
Yeah, I finally got my winter coat out for a change. And I walked outside and I was like, whoa, wait, I'm not actually freezing for once.
Okay, I hope Dane joins us soon. Okay, first of all, great job yesterday. Thought you guys worked really well.
Jenna and I have been lab partners since like freshman year.
Well, yeah, go ahead. It's amazing how much trouble people have following protocols sometimes. And I had a class once where it said on the protocol, add 2 microliters of reverse transcriptase. The students took the reverse transcriptase, which is an expensive enzyme, and they added 10 microliters to all their tubes. We're out of reverse transcriptase. That's just, guys, read your protocol.
It says add two.
Yeah, so Anyway. The way that works, so anyway.
No, when I was in my internship, a lot of times I didn't even have official protocol to go off of. My boss would just like write chicken scratch on a piece of paper and be like, "Here's the protocol." I was like, okay, I guess.
Yeah. That's just painful.
But everything, apparently I did everything right, so. I didn't play too much. Update on the Johnson & Johnson vaccine.
The sweater makes me kind of feel like a flying squirrel.
You do look like a flying squirrel. And then I can put popsicle.
It's scary how much we're like.
I always do terrible in these quizzes. RIP.
Okay, question 2, I should have worded that differently. Which of the following is not a bike path? There's only one answer to that.
Oh, okay.
Makes it sound like there's more than one answer. There's only one.
R.I.P.
So many times I was like, I don't remember this, but I don't remember the answer.
That's what you're supposed to study.
I did. There was one of the questions that I was like, I literally studied this exact question. I found it, and then I just could not remember the answer. And I think I did the exact opposite. I didn't do that.
I have no idea.
See if you did any better than I did.
Probably not. I probably did worse.
I doubt it.
So the binding of glucagon to its receptor increases the intracellular concentration of cyclic AMP.
Dang it, that was gonna be my second choice.
I got one right.
Cyclic AMP.
I remember that was the one question I found, and I found it on like Khan Academy, and I guessed it, and I got it wrong the first time, but then I forgot what the right answer was. So I think I selected the same one that I guessed the first time.
Both of you got the second one right. That reaction is catalyzed by aldolase. The aldolase reaction is reversible. It's not a bypass reaction. The rest of these are bypass reactions. The enzyme that catalyzes this is UDP glucose pyrophosphorylase. Pyrophosphorylase. Glycogen synthesis starts with glycogen. Glycogen, which you got right.
I didn't remember how to spell it. I was like, is it?
Positively regulated by insulin. Because insulin favors the synthesis of glycogen, right? Which is not a substrate for protein kinase A, glycogen phosphorylase. The rest of those are. That's the first quiz. There will be others. Dane is having trouble getting in, I think.
I just realized I have a problem and I have to like come up with it without like the enzyme of like, oh.
Oh, he's in the waiting room? Let's see, waiting room.
I think like the participants that might be in there.
Um, oh, good gosh. Default email.
If you go to the, instead of the area you click for participants, click the actual participants, yeah, and see if that. Okay, that would tell you if who's in the meeting.
Yeah, I would. And no,
He says he's in the waiting room, he's in a different.
he's in a different one, which leads me to believe that the. Okay, I'm just gonna go forward. There he is.
Yeah, there we go.
Sorry about the runaround, buddy.
No, you're fine. Don't worry about it. Technology is great when it works, right?
Yeah, when it works. Okay, I'm gonna show some videos. I don't know if you'll hear the video well enough, but I think my microphone will pick up the speaker right there.
Uh-huh.
So we'll do the best we can with what we have.
Okay.
All right, let's review. So we see that the active site of our Rubisco, first of all, requires this magnesium, have to have the magnesium. Can't do this without the magnesium. You have these two consecutive acidic residues, the D and the E, and those are absolutely conserved in all Rubiscos, be they in photosynthetic bacteria or mosses or green plants or Euglena or whatever, those are conserved, absolutely conserved. You have this lysine that's usually nearby. It's a 201 in green plants. Sometimes it's a little closer, a little further in others, but you have a lysine. That lysine is carbonylated in the CO2. That's a regulatory step. These will hold magnesium in place. The magnesium holds the two substrates, ribulose 1,5-bisphosphate, and the CO2 in place for proper attack of one on the other, so that you can make the 6-carbon unstable intermediate. So again, we went through this. Let's review it very quickly. The first substrate that binds is ribulose 1,5-bisphosphate. You can see that it is held in place very nicely by the magnesium. You get abstraction of this hydrogen here by the carbonylated lysine at 201. Then you have a double bond here. That's your immunodilate intermediate. That is the subject, the picture that's attacked by the carbon dioxide. Carbon dioxide will attack here, you form a bond between this carbon, that carbon. This is your 6 carbon beta-keto acid intermediate. That's very unstable. Now water. Water will come in and it will make a even more unstable hydrated intermediate. And then as you can see, this is going to, this upper piece here is going to take its electrons and leave. That's your first 3-phosphoglycerate that leaves. This then is going to recapture a hydrogen from the nearby lysine below, lysine 201. In green plants, it's lysine 175. In others, it's 180, 172, but it's there, it's below the 201. Then this gives the hydrogen here, and this hydrogen that was abstracted goes back down here to regenerate that. This is released. That's the second substrate that's released. This should remind you of a mechanism. This is a ping pong mechanism. One binds, other binds, one substrate's released, the other substrate's released. Very much a ping pong mechanism.
So there's 2 individual 3-phosphoglycerides.
Yes. Yes, and one stays in the stroma, the other leaves, which we'll see today. So, and then the summary of this is you get 2 3-phosphoglycerates from your lowest 1,5-bisphosphate and CO2. That's what Rubisco accomplishes. The rest of this will show you how you use this for gluconeogenesis and how you regenerate rigorless 1,5-bisphosphate. You must regenerate this, or this process stops in its tracks. Very important. All right, questions before we go on?
For the overall theme, does it use your, sorry, the Rubisco is used if gluconeogenesis just needs more phosphoglycerate, 3-phosphoglycerate, or
Well, this whole process, as we're going to see, is very expensive energetically. Requires a lot of ATP, requires a lot of reductant. When in plants do you make ATP and reductant? When the lights are on. So even though these are called the dark reactions, they occur when the lights are on. They must, because you can only do this when you're making ATP and reductant. And that's when the lights are on. And we'll see the way this is regulated is in a light-dependent fashion, which is, that's unique to plants. You don't see that in animals. So Rubisco on the stroma, and it is very heavily expressed in the stroma.
is that expressed in everything or mostly plants?
Plants. Animals don't have this enzyme. It's a plant-specific enzyme, and it's expressed in the chloroplast. You don't have it in the stroma. You don't have it in the cytosol. It's in the stroma, which is the soluble bit of the chloroplast.
So Rubisco only has what it needs when the lights are on.
Yeah. And in fact, let's go to the regulation of it. So the carbamylation of lysine 201 is catalyzed by Rubisco activase. Rubisco activase. Rubisco activase will make sure you get this carbamylation when you need it. Then some plants use a transition state analog to inhibit rubisco in the dark. And this is the 2-carboxy-arabinitol 1-phosphate. If you look at that and you look at ribulose 1,5-bisphosphate, you can see some similarities structurally. So that is a transition state analog. And this transition state here looks a bit like that transition state inhibitor. All right, so here is the active site of rubisco. So it binds the ribulose 1,5-bisphosphate. It's inactive. You must have this epsilon amino group carbamylated in order for it to be able to execute its function. So the rubisco activase uses ATP to displace the ribulose 1,5-bisphosphate. Then CO2 will actually carbamylate this in a non-enzymatically activated fashion. When you carbamylate this, you can bind the magnesium. Only after you carbamylate that lysine 201 can you bind the magnesium. Otherwise, magnesium binding is just too weak. So the activase gets this bound ribulose 1,5-bisphosphate out of the way. Because if this is bound, there's steric hindrance of that carbamylation.
This might be a dumb question. Carbamylation, that means adding carbon.
You add CO2 and you make a carboxylic acid group. That's carbamylation. And if you want to pronounce all the syllables, you could say carbamylation, but I kind of slur it through because I think carbamylation sounds really stupid. So I say carbamylation and just slur it through. It's much easier to say. And my jaw doesn't hurt talking about it. All right. Oh, okay.
We're in America, we're allowed to do really long words.
All right, now, so the 3-phosphoglycerate will be converted into glyceraldehyde 3-phosphate in 2 steps. Glyceraldehyde 3-phosphate. Where have we seen that? Krebs cycle. Sorry, glycolysis. This is material you can use for gluconeogenesis. So here are the 2 steps. They're shown here. So first, you phosphorylate this with a 3-phosphoglycerate kinase. Then you are going to reduce it with a glyceraldehyde 3-phosphate dehydrogenase. This is an NADPH using enzyme that makes it different from the glyceraldehyde 3-phosphate dehydrogenase you might find in the cytosol, which uses NAD. That gives you glyceraldehyde 3-phosphate. And now you can keep it in the cytosol, or you can get rid of it to the, keep it in the stroma or get it into the cytosol. Now, if it goes to the cytosol, you'll use it to make sucrose and it will be transported from the cell.
It makes starch.
Yes, stays in the stroma. It's used to make starch. to make glucose, and then from that glucose, you make starch.
Did you say, so glyceraldehyde 3-phosphate goes to glycolysis?
Gluconeogenesis.
Gluconeogenesis. And what is glycolysis at the top part saying? The very, very, very bottom of the page. Yeah.
Oh, if you want to make ATP from this can go through glycolysis to make ATP.
Oh, okay.
Usually you're going to do that. If you're photosynthesizing vigorously, you have plenty of reductant and ATP, then you'll use this to make hexose sugars. There's an experiment you can do with chloroplasts. You can take chloroplasts, say from spinach, which are pretty easy to isolate. You can then shine high intensity blue light on them, and you'll see very robust starch granules. Then you can put them in the dark for several hours at low temperature and come back, and the starch granules will be gone. They were used essentially as a energy source. So that shows you how when the lights are on, you make starch. When the lights are off, the starch is degraded.
And this isn't like full spinach plant.
Yeah, you pick the spinach leaves and then you isolate chloroplasts, which there are kits to isolate chloroplasts is really easy to do. And then you can actually do experiments if you put them in the right buffer. You can do experiments with the isolated chloroplasts. And they will act very much like chloroplasts in mesophyll cells.
So I'm curiosity, are all of these irreversible?
These reactions? No, because when the plant is in the dark, starch is degraded and the sugar is degraded by glycolysis to make ATP for the plant because there's no light. That's why plants, yes, they make oxygen when the lights are on. When the lights are off, they make CO2. All right, these two steps then. These will make glyceraldehyde 3-phosphate through a phosphorylation and then a reduction. And this glyceraldehyde 3-phosphate can be converted to dihydroxyacetone phosphate by triose phosphate isomerase. Yes, by a glycolytic enzyme, but there is a chloroplast specific isozyme for it. That then converts it to dihydroxyacetone phosphate, and a aldolase will then combine these together to make fructose 1,6-bisphosphate. From there, you know what happens. And then it goes through and it's used to make starch. And we went through starch synthesis already. Okay. Okay, how do we regenerate ribulose 1,5-bisphosphate? Some of the glyceraldehyde 3-phosphate stays in the chloroplast in order to be converted to starch. Some of it will go to the cytosol, we used to make sucrose. There's this phosphate, triose phosphate antiporter that will take your dihydroxyacetone phosphate and transport it into the cytosol for gluconeogenesis and conversion to sucrose. like right here, that shows you this here. And this whole thing, as you can see, is powered by photosynthesis. All right, one stays, one goes. One goes to cytosol, the other will stay to regenerate ribulose 1,5-bisphosphate. Now you're going to employ the non-oxidative reactions of the pentose phosphate pathway. There are two key enzymes that the cell will use. These enzymes are transaldolase and transketolase. Transaldolase and transketolase. And here's how you do it. And if this looks complicated, that's because it is. So this is the grand scheme. Let's go through these enzymes here, how they work, then we'll go back to this. All right, so the two main enzymes. Transaldolase will catalyze the condensation of a ketose sugar with an aldose sugar. A ketose sugar with an aldose sugar. It condenses them together in an aldol condensation to make a bigger sugar. So you start with smaller sugars, you make bigger sugars.
You said start small, go big.
Yeah. So if we back up, let's take a look here at this reaction here. We have a ketose compound here, dihydroxyacetone phosphate. We have a tetrose over here, an aldotetrose erythrose 4-phosphate. One is a ketose, the other is an aldose. Transaldolase condenses them together to make the 7-carbon compound sadoheptulose 1,7-bisphosphate. Sadoheptulose 1,7-bisphosphate.
So for this, we don't necessarily need to know the structures, we just need to know how many carbons there are in this one. You know, the structures for this.
You need another structure. So that's dihydroxyacetone phosphate. And then erythrose 4-phosphate. So you take your ketose, you take your aldose, and your transaldolase. This has a lysine group, and it uses a Schiff base mechanism. A Schiff base mechanism. And you're going to get
you just pause for a minute? So I can
Yeah,
get
pause for a minute.
Is the,
Acetoheptulose 1,7 bisphosphate.
is that fourth carbon down? Is that another OH to the left?
Yeah,
What does
oops, oops.
What does the
oops,
TA
oops.
stand for again?
Transalloylase.
I was like, what?
And the acetoheptulose is what is formed by the shift base mechanism.
Thing up there, we have 2 3-carbon compounds, and then you make fructose 1,6 bisphosphate. So it's the same thing. You have a ketose sugar, aldose sugar, you combine them with an aldol condensation reaction to make a larger sugar. All right, the second enzyme that is used extensively is transketolase. Transketolase. This will take a ketose donor and an aldose receptor. You're going to give this ketose a 2-carbon haircut. You take this and move it to here. You're going to get an aldose receptor that is now 2 carbons larger, and ketose donor is now 2 carbons shorter. And you go, zaba boom, there you are.
What's the name of the really big compound again that we make using?
Acetoheptulose 1,7 bisphosphate, it's right here.
Okay. I'm gonna copy it. We can move on. Thank you.
Just a word of this particular enzyme uses a thymine pyrophosphate cofactor. This enzyme is very, very prevalent in red blood cells. And in fact, you can take blood and you can do a transketolase assay. And if the activity of transketolase is low, that is diagnostic for a thymine deficiency. That's why it's so easy to test for thymine deficiency.
How common are thymine deficiencies?
In this country, very low, because our food is fortified with thymine. In countries where the main diet is polished rice, then thymine deficiencies are much higher.
Are you just swapping the R groups then?
Sorry?
Like, in the reaction, is it basically just cutting off the R groups and?
You cut this off here. This then will become an aldose. This then will become a ketose. This is an aldose and it's 2 carbons shorter. This is now a ketose and it's 2 carbons larger. So this particular ketose donor is turned into an aldose. It's 2 carbons smaller. The aldose receptor is turned into a ketose and it's 2 carbons larger. All right, so backing up again, take a look at where we have transketolase. Let's look right here, this reaction here. Fructose 1,6 bisphosphate, sorry, fructose 1,6 phosphate and glyceraldehyde 3-phosphate. Aldose, ketose, 6,3. You're going to take these 2, put it here. This will now be 4, that'll be 5.
So transketolase is used multiple times in this process?
Yes, and so is transaldolase. So this then becomes erythrose 4-phosphate. This becomes xylulose. It's a ketose 5-phosphate.
So is it safe to say that like transaldolase combines?
Yep.
And transketolase kind of just moves?
Yeah. Yeah. In fact, if you want, if it helps you, as you draw this out, you can maybe paint these 2 dots here. You can paint them red. And then
And then
paint
the
these 2 red. So you know they came from here.
Is it always moving two carbons? Is it
Yep.
having two?
Always moves two. And it always moves two from the ketose,
Okay.
always.
And it's always in aldose that it's working with them too.
Yeah. It's always transferring the top carbon and the ketone group to the aldose. All right. So these reaction schemes start with 2, 3 carbon compounds, which makes sense because that's what you start with. Then you go a 6 and another 3. And you're going to do a transcatolase reaction here. That's a transallylase reaction. Then you go from the 6 and the 3. Now you have a 4 and 3. Then you combine the 4 and the 3, you have a 7 and a 3. That sounds like a transketolase reaction that you're going to get. And now you have a 5 and a 5. You've now formed 2 5s.
So it goes, I'm just gonna test myself. So the first one is transallylase.
Yeah, and let's go back to here.
Oh, so it's this. Just
Right,
one
so
down.
transallylase. Then you get rid of the bottom phosphate. Fructose 6 phosphate, now you have a 6. Then you go 6 and 3. That gives you 5 and 4.
Then 4
5 and 4. Then 4 and 3. This is a 5. Good. All you need to do now is use that epimerase. The xylulose is an epimer of ribulose. Then all you need to do is phosphorylate that. You have ribulose 1,5-bisphosphate.
Okay, so do those happen at the exact same time?
Yeah.
Okay.
Yeah.
So how many ribulose 1,5-bisphosphates are you getting out of this?
You're going to need, you're going to make a sugar. You're going to need to make 5 of them in order to regenerate ribulose 1,5-bisphosphate.
Okay.
So you need to kick, you fix 6 carbons. Out of those 6 carbons, 5 of them are used to regenerate ribulose 1,5-bisphosphate because it's a 5-carbon compound.
Is the numbers the order of how it happens?
Yeah. Yeah. And from this, get rid of phosphate, because if you have that phosphate up there, you can't use transketolase. So then you go 7, 3, then you go 5 and 5. Xylulose, well, just use the epimerase, ribulose 5-phosphate, phosphorylate it. Well, first convert it to ribulose, because this kinase only works on ribulose. Then you go from here to there, and you got one there, 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 and you made it.
So you have 3 glyceraldehyde 3-phosphates coming in and 2 ribulose 1-bisphosphates coming out.
Yeah, yeah,
Okay.
Yeah.
Making sure
yeah. Well, actually, yeah, 3, right, exactly. 3, so 3 times 3, 9. If you want to make 3 of these, that's 15. You'll need 5 of these that eventually come in.
Okay.
Yeah, we'll need-
Really
Yeah.
quick question. What's the difference between, so you have the ribulose 5-phosphate, you have the epimerase, the kinase, and the isomerase, right, of all 3?
Yeah. Yeah. Also, Amanda, keep in mind, these come from glyceraldehyde 3-phosphate, don't they?
Yeah.
So it's 1, 2, 3, 4, 5. 5 times 3 is 15. 3 of these. 3 times 5 is 15. So the carbons count up, don't they?
Oh,
Okay.
gotcha.
That makes so much more sense.
Yeah. Yeah.
It's a lot, but it makes sense.
Yeah. Okie dokie. Alrighty, dokie. Let's see if there's any questions. Okay, good. And here you can see the isomerase, the epimerase, and the kinase. The ribose 5-phosphate isomerase interconverts between ribose 5-phosphate and ribulose 5-phosphate. The xylulose 5-phosphate is converted to ribulose 5-phosphate by the ribose 5-phosphate epimerase. And then the ribulose 5-phosphate kinase converts ribulose 5-phosphate to ribulose 1,5-bisphosphate. And so glyceraldehyde 3-phosphate or dihydroxyacetone phosphate can enter at several different places. The net result is you regenerate ribulose 1,5-bisphosphate. Now you saw the ATP you had to put in to do that. This process takes ATP. So each triose phosphate you make requires the expenditure of 9 ATP and 6 NADPH. It doesn't directly require light, but it requires ATP.
So it will only happen when there's light. Because
Right, so it only happens when there's ATP, which means it only happens in the light.
Wait, sorry, can you go back? So you said it requires ATP.
Right,
therefore it kind of-
which, yeah. So it doesn't directly require light. So it's used to make sucrose for transport, it's used to make starch for storage. The energy generated by photosynthesis will be used to make those 3 carbon compounds that are then used to make starch and sucrose. And these occur in the stroma of the chloroplast. We'll see that photosynthesis occurs in the stacked thylakoid membranes of the chloroplast. Yeah.
Why are they called the dark reactions if they occur when they're-
Because when Kelvin discovered them, if he added ATP, he could get the reactions to occur in the dark.
Oh.
And because they didn't require light, they were called the dark reactions. That's a little deceiving though, aren't they?
Yeah, okay.
Because they occur during the light.
And do all dark reactions occur during the light?
Yeah. Yeah.
I don't like that.
Yeah, I don't like that. That's why I call them the carbon fixation reactions or something like that. Because that dark reactions is classical, but it's bloody confusing.
So the only way it can happen in the dark is if you like artificially add ATP.
Yeah, and that's what Calvin was able to do. So he covered the windows of his lab with foil and then put his experiments under a shoebox. And he discovered, then he took assays. He kept doing assays. He'd pick up the shoebox, do the assay, and he found they still made glyceraldehyde 3-phosphate, even though the lights were off.
I feel like that's not a fair experiment if you didn't allow it to happen naturally.
Well, that's why they got called the dark reactions. So Calvin, boy, give him kudos for ingenuity and doggedness.
Yeah.
All right, so Rubisco is subject to, first of all, the regulation by carbamylation. Carbamylation is probably the most important mode of regulation for Rubisco. Then it's subject to transition state regulation by the 2-hydroxyrabinitol 1-phosphate. Then the other enzymes of the Calvin cycle are subject to light-based regulation. There's 3 different light-based means of regulation. Let's go ahead and look at these.
So these are different because they have to happen.
Yeah. You know what? I got this wrong. Sorry. No, that didn't look right. Sorry. Now, when the lights are on, plants are pumping protons from the stroma into the thylakoid sacs. Therefore, the pH of the stroma goes up during photosynthesis. Some of these enzymes have their pH optima in the alkaline pH range. And when the lights are off and the pH of the stroma falls below this alkaline pH, they're not very active anymore. That's a first mode of regulation. Second, photosynthesis releases magnesium into the stroma. And several of these enzymes have magnesium cofactors that are required for their activity. So that magnesium is only available during photosynthesis. So let's show you fructose 1,6-bis phosphatase. Fructose 1,6-bis phosphatase requires magnesium, and look at the pH optimum of this. pH 7, which is pH you get when the lights are off, it's not very active. Also, its magnesium tends to be taken from it, in which case it won't be active at all.
Can you repeat the correlation between magnesium and pH?
Magnesium tends to be bound by the membrane when the lights are off. During photosynthesis, the magnesium is released and is available to these stromal enzymes now.
So you're saying that some of the enzymes have to have magnesium. So
Yeah.
if they don't have it, so
They're not
so-
active.
okay,
Right,
so
it's a
because they're
cofactor.
bound During, well, not during photosynthesis, then when photosynthesis occurs, these enzymes can work.
During photosynthesis, these enzymes work when the lights are off, pH is too low for them to work effectively, and then the magnesium gets sequestered. So that's a first mode of light-dependent regulation. Secondly, when you generate reductant, you make the chloroplast a reducing environment. When the lights are off, the stroma now becomes an oxidizing environment. So many Calvin cycle enzymes have disulfide bonds. Those disulfide bonds must be reduced for them to be active. When are they going to be reduced? When there's reductant around. When there's reductant around? When the lights are on. So these 4 enzymes are all involved in the Calvin cycle. All of these have cysteine residues that participate in inactivating disulfide bonds. When they're reduced, the enzymes are active. When they're oxidized, the enzymes are inactive. When they're reduced, when the lights are on. When there's reductant. When are they oxidized? When the lights are off. And then these enzymes are not active anymore. So fructose 1,6-bis phosphatase, acetoheptulose 1,7-bis phosphatase, glyceraldehyde 3-phosphate dehydrogenase, and ribulose 5-phosphate kinase.
Does this only happen during the Calvin cycle, or are all of these turned off?
In the chloroplast, these are off when the lights are off, because they're tied up in disulfide bonds that inactivate them. And so there's a ferroredoxin. That ferroredoxin is reduced by photosynthin 1 in the presence of light. That reduced ferrodoxin will then, as you can see here, will reduce this thioredoxin. And then the thioredoxin, with the help of the ferrodoxin thioredoxin reductase, the thioredoxin will reduce these enzymes. In this state, the enzymes are inactive. In this state, they're active. In the dark, the enzymes are in this state and are inactive.
Are you still watching those videos today?
Yeah. Okay. Okay, let's
The leaves of this plant have cells that carry out photosynthesis. If we zoom in on this photosynthetic plant cell, we can see the chloroplast, where the reactions of photosynthesis occur. Photosynthesis consists of two primary steps, the light reactions and the Calvin cycle reactions. In this tutorial, we'll focus exclusively on the Calvin cycle reactions. The overall purpose of the Calvin cycle is to convert carbon dioxide from the atmosphere into carbohydrates or sugars, which the plant needs to power its cellular activities. The Calvin cycle is divided into two phases. Carbon dioxide
okay, that's ricochet science and the Calvin cycle. All right, it is time to end. We will finish this on Monday, and then we will get into the
Let's talk
synthesis
about one
of
the most
fatty
important
acids.
biological
No, sorry. Take a look at cell wall synthesis. That's another way you make polymers. Look at cell wall synthesis in plants and then also in bacteria. Okay. Dane, any questions? No, you're good? Okay, stay well. You?
Whole idea is plants and not just bacteria, algae, other things, but we know.
Yeah. Okay.
I'm so proud of us. We got the lights on.
Woo!